Great. Thank you very much, Nick, for the introduction. So obviously, for people like me, a cardiac radiologist, UK Biobank has been completely transformative in what it's enabled us to do, particularly around understanding the genetic regulation of complex traits and of prognostically important aspects of cardiovascular health. But I think the future opportunities will really lie in imaging, leveraging the potential of multiomics, of non-imaging phenotypes like ECGs and genetics. And I think that can partly be achieved by working on more realistic models of the cardiovascular system that are potentially closer to the pathobiology of disease, and also developing machine-readable models that potentially can integrate lots of diverse data sets. And I think that will help us towards moving towards impact, towards target prioritization, towards making individualized predictions about people's health and trajectories, and towards preclinical validation. And I think, as some of the other talks have touched on, I think a real value in the future will be, once now we've done all 100,000, is to be able to integrate imaging with other data sets, brain and whole body imaging as well. But also to leverage the value of multi-omic data sets, and as Nick has touched on using the longitudinal data to be able to look at life course trajectories and aging. So just going to touch on 3 use cases around organ development, around determinants of cardiovascular aging, and on gene disease discovery and drug therapy. So you've heard from Wenja and others, it wasn't really given that it would be possible to be able to analyze the cardiovascular images at scale. And through the great work from Stefan Peterson's team and Wenja's team, we're now able to derive image-derived phenotypes at scale on the whole 100,000 data sets. And this isn't a biobank person, but it gives you an idea of how these images can be converted into digital form by labeling each pixel in the image according to their anatomy. And I think this is roughly where we're at now in terms of building these more realistic models of the left ventricle. This is data aggregated from 20,000 people in UK Biobank. And this shows the effect of carrying titin variants, which are associated with heart failure, on the function of the heart. So these are cases versus controls, and you see the complex patterns of motion that these variants are associated with. And also, as Patsy's touched on, having these 3-dimensional models allow us to look at regional changes. And for instance, HCM, leading cause of sudden death, is a very regional disease. So having these 3-dimensional maps of wall thickness allows us to discover new loci related to cardiomyopathies. We see in green here, if we do this spatial GWAS, we can see all of these number of new loci, many of which linked to cardiomyopathic diseases. Now, one of my interests is around trabeculation. So these are the fine muscular strands that line the inner surfaces of the heart, and they're conserved across the whole of vertebrate evolution. And they're certainly important in the developing heart, but it's really been unclear what they do in the mature adult heart. And they're probably doing something important, being somewhere as critical as that. But again, UK Biobank allows us to start to answer this question, first of all, by finding ways of phenotyping them. And we can do edge detection, essentially, to find the boundaries of these trabeculae, and then do fractal dimension analysis to basically give us a number of how complex these trabeculae are. And then that fits into a workflow of phenotyping in Biobank, developing a trait, doing genetic association analysis, which tells us about cytoskeletal development in the heart, going to knockout models, moving on to outcome studies, and also using human-informed bioengineering models that we can perform experiments on. And just to give you a flavor of this, this is a Medaka fish. It's an experimental model. And one of the genes we discovered associated with trabeculation is MTS1. You have the wild type at the top, and you can still see these branching trabeculae. But if you knock out that gene, you end up with a smooth endocardial surface. Interestingly, I think, previous work had shown that if you knock out that same gene in developing nerve cells, you also get a loss of this arborization pattern. So potentially, nature's found a way of increasing surface area across different organs using the same sort of pathways. And more recently, we've been extending this to the larger data sets that are now available in Biobank to look at both common around the outside of the circle and rare variants on the inner side associated with trabeculation. And these point to potentially these polygenic scores for trabeculation being an important modifier of the cardiomyopathic phenotype and explaining some degree of the diversity of cardiomyopathy phenotypes that we see in the clinic. And actually, in a way, I think this kind of points to some of the problems that we're trying to solve is understanding the interaction between those common variants, those pathogenic variants, and environment, particularly blood pressure, diastolic blood pressure, and HCM, how all of those interact to modify the phenotype. And you have to understand each of those to be able to make an understanding or prediction of the disease phenotype. Aging, of course, is one of the major risk factors of cardiovascular disease. We have examples of young and old hearts here. And you can train a machine learning model based on dozens of imaging parameters to be able to predict someone's biological age. And then the gap, as we've heard before, like your age delta, is your deviation from healthy aging. More positive, the worse. And that, of course, could be due to polygenic risk, early life factors, later life exposures, for instance. And we can use age gap to look at some of the genetic variants associated with aging, do something similar with ECGs. And some of the key messages are around pathways that regulate atrial fibrosis, stiffness of the vessels and the heart muscle, but also inflammation as well, pathways that regulate monocyte macrophage inflammation. And of course, one of the key things that we know is pro-inflammatory in the body is fat. And this touches a bit on some of the messages we've heard earlier, but actually integrating both cardiovascular aging and assessments of body fat distribution allow us to show important sex differences. Males particularly very sensitive to visceral fat in terms of aging, but gynoid distributions of fat and estrogens appear to be relatively protective. So important sex-related differences in drivers of aging, probably because of the pro-inflammatory and pro-fibrotic effects of fat. Now, I touched about how imaging can maybe enhance the value of other data sets. So knowledge graphs are a very powerful way of representing and organizing information in a machine-readable way, using knowledge gathered from many databases representing the relationship between diseases and genes and so on. And if you add imaging as a precision endophenotype that's really close to the biology and close to the disease, then you can potentially really enhance prospects for drug discovery, drug repurposing, and understanding gene-disease linkages. So here is imaging, cardiac MR sitting in the middle of this complex knowledge graph, integrating over 18 different biomedical databases. And again, we can train a machine learning model then to be able to predict new gene disease associations, what drugs might target those genes, and what drugs could be repurposed as well as a relatively cost-effective approach for treating diseases. So potentially a very powerful way for understanding new approaches for treating people, where imaging really leverages the value of these other data sets. So lastly, I just wanted to touch on the collaborative nature of Biobank. I think it spans all of clinical imaging and machine learning, genetics, preclinical models and therapies. And I think through these collaborations and through the future work towards the 60,000 imaging party we're going to have at some point, I think it will be hugely impactful that what UK Biobank delivers for cardiovascular health. Thank you.